Hello everyone. In this video, we will be talking about the physiological mechanism of tDCS in treating major depressive disorder. Several studies and meta-analyses have demonstrated tDCS as a safe and effective treatment for depression. tDCS is a non-invasive method that modulates neural activities in the brain by delivering low amplitude of no more than 2 milliampere for 30 minutes in 10 to 15 sessions. In the image, we can see the anode and cathode electrodes of 35 centimeters square in size at point B and C, positioned over the left dorsolateral prefrontal cortex and over right supraorbital region, respectively. The direction of current flow is from the anode to cathode, that is from positive to negative, to facilitate or inhibit spontaneous neural activities in the vicinity of electrodes. Depression is a condition related to dysfunction in several cortical and subcortical areas, especially the dorsolateral and ventromedial areas of the prefrontal cortex, the amygdala, and the hippocampus, which are associated with symptoms such as psychomotor retardation and executive dysfunction, as well as feelings of hopelessness, etc. Patients with MDD have very low activity in the brain, as shown in the image. MDD is hypothesized to be the result of monoaminergic and long-term potentiation deficiencies. It is considered that one or more of the monoamine neurotransmitters, for example serotonin, for one reason or another, become less able to carry nerve signals across nerve junctions called synapses. It is generally assumed but remains unproven that the problem is due to the shortage of neurotransmitters. Nevertheless, any impairment consequently results in cortical subcortical dysfunction, which is also reflected in greater HPA activity. However, there is strong evidence that TDCS affects neurotransmitter concentration. Long term potentiation is an important process in the context of synaptic plasticity. Any impairment can explain depression, as depressed subjects present deficits in declarative memory consolidation, enhanced fear acquisition, and impaired visual discrimination, suggesting deficits in LTP mechanisms in the hippocampus, amygdala, and the visual systems. The neural stimulation over the left dorsolateral PFC has shown improvement of executive function in working memory tests, as well as enhancement of identification of positive visual material. Nodal stimulation with TDCS by itself is not strong enough to depolarize the membrane potential of neurons to the firing threshold and only increases the rate of spontaneous combustion and their excitability. Conversely, cathodal stimulation is thought to deepen the resting membrane potential, that is, hyperpolarize, making it difficult for neurons to depolarize, which reduces spontaneous combustion rates and excitability of neurons. Importantly, these effects of TDCS depend on the intensity and duration of stimulation and radial electric field. Actions on NMDA receptors inducing plasticity of neurons play a dominant role in improving symptoms of depression. This is because NMDA receptor is involved in the intracellular influx of calcium ion during depolarization, reduces prolonged action potentials, and mediates neural circuits governing memory and learning. The nodal stimulus also causes a local decrease in GABA concentrations in the cortex. Moreover, monoamine neurotransmitters such as dopamine, serotonin, and acetylcholine have been reported to mediate the effects of TDCS. It modulates the dopamine system, enhances and suppresses serotonin and acetylcholine transmissions, respectively. Hence, the direction of influence of actions of a nodal stimulation varies depending on monoamine neurotransmitters. Continuous enhancement of signal transduction between neurons is thought to mediate the effect of TDCS. First, action potentials in presynaptic neurons are converted into chemical signals at the presynaptic membrane. Subsequently, neurotransmitters are released into the synaptic gap, and this process is called the signal transduction cascade. In this cascade, various neurotransmitters either activate or inhibit transduction cascades bound to G proteins or ion channels, leading to phosphorylation of CREB and activation of genes in the nucleus of neurons. Also, the neurotropic factor-bound transduction cascade may play a role by activating various kinase enzymes. These signal transduction cascades enhance the synthesis of various proteins, such as neurotransmitter synthesis, receptors, ion channels, and intracellular signal proteins. Facilitative actions of these proteins that regulate efficiency of neurotransmissions in the cerebral cortex circuit may explain the ability of tDCS to induce LTP. tDCS can elicit antidepressant effects, however, it has not been demonstrated as superior to standard first-line antidepressants in terms of overall efficacy. However, benefits include its minimal side effects and low risk for adverse events, low cost, and accessibility. Nevertheless, optimization can occur by manipulating dose, including electrode size, configuration and charge, combination of pharmacological or cognitive therapies, refining electrode placement, and stimulation based on individual structural and functional anatomy and connectivity. Understanding of how electric fields penetrate the brain to engage and modulate particular neural circuits will also advance efforts to tailor treatment. tDCS elicits antidepressant effects in both unipolar and bipolar depression, but is less suited for treatment resistant patients. Research also suggests tDCS may better target dimensions of depressive illness and that patients with particular pretreatment symptom profiles will have a greater response. At present, measures of gene and immune system function have shown fewer links with tDCS related clinical response, but few studies have addressed this question. Future research, including imaging guided tDCS, more comprehensive clinical and cognitive phenotyping, and data driven approaches to identify more salient predictors of response, are still needed. Thank you. Here is the list of references.